Hi there, in this tutorial I'll show you how to look up data on your favorite gene using GeneProf. So I'll just enter NaNoC as a search term in the search box on the main page of GeneProf. That's one of my favorite genes. And GeneProf presents me with a page of a lot of search results. There are genes and experiments from various organisms connected to my search term. So the first thing I'll do is I'll untick experiments and select only mouse to narrow down my search. There's another tutorial available just on searching, so full details available there. So anyway, after I do this, I get only two results. That's the NaNoC gene that I'm interested in and another pseudo gene. And I click on the main entry. This will open up the main gene page for NaNoC. And we find on this page at the very top some generic information about the gene, just like the name, the description, the type of feature we're looking at. There's a protein structure mined from the PDB database, some generic information about the genomic structure of the gene, various exons that it consists of, and transcript variants that are known. There's also a little diagram depicting this. So we can see that NaNoC, for example, consists of 1, 2, 3, 4, 5 exons, which are connected by various intronic regions. Next, we can also find some functional annotation that's mined from the gene ontology. We can see that the NaNoC gene is involved in stem cells and developmental processes. There are a whole lot of protein-protein interactions known for this gene from a variety of different studies. These have all been mined from the BioGrid database. And finally, there's a section with identifiers in external databases and link outs to these databases. So you can go, for example, to the Ensemble database and look up the corresponding gene entry in that on their website. Now, more interestingly, really, below all this general information, there's a section detailing the actual GeneProf specific data. So you can see, for example, first, there are 3 experiments in the GeneProf database that specifically deal with NaNoC. And then next to it, there's some information about the expression of NaNoC based on the data in the GeneProf databases, on all this RNA-seq data that we have available. You can see that NaNoC is most strongly expressed in embryonic stem cells, also in iPS, these are induced pluripotent stem cells, and its expression comes on as early as at the 4-cell stage in the plastomere. You can choose to summarize the expression data in other terms, like in tissue, or treatment, various other annotation terms. And full details about all the numbers where this plot is based on are available in an expanding panel above. So you can see a list of all the experiments that provide data on the expression of NaNoC. You can annotate them with various categorical terms, and the actual numbers are available there. You can pick values for experiments that we're interested in, and then browse this data together with data on all the other genes in a dynamic table browser. There's another tutorial available that tells you exactly how you can do this. So let's go back to the gene page. Next, after the gene expression data, there's a list of strongly correlated genes. So these are genes that, based on their expression profiles, are highly similar to NaNoC expression. Below that, there's a section that deals with the transcription factor binding activity of NaNoC. NaNoC itself is a transcription factor, and there are 3 different datasets in GeneProf database that list putative target genes of NaNoC. You can open that again in a dynamic table, and here I filter for genes that are found as potential targets in all the 3 different experiments. There are almost 3,000 of them, and find that it, for example, contains the stem cell transcription factor POW5F1, a known target. And back on the gene page, there's a table of the 25 most strongly bound target genes based on, again, the average of these 3 studies. You can again open a table containing the full data. And the final section of the page contains transcription factors, DNA binding proteins, and histone modifications for which an enriched activity has been observed somewhere in the proximity of the nanopromoter in at least one of the experiments in the GeneProf databases. You can see that it contains, amongst others, the stem cell transcription factor POR5F1, and I'll just tick the checkboxes behind some random datasets in this list here. So SOX2, another transcription factor, a histone modification H3K4ME3, and some POL2 ChIP-seq data. I'll tick the checkboxes next to the genomic datasets there, and click the Browse button at the very bottom of the page, which opens up those genomic datasets centered on the nanoclocus in the genome browser. You can see very nicely that the 2 transcription factors, the histone modification, they've all show an enriched binding directly at a nanopromoter. The transcription factors have another enriched event just about 5 kilobases upstream of the promoter, and the POL2 binding data also shows activity at throughout the nanogene. Okay, this is all I wanted to show you for this session. I hope you enjoyed this, and I'll see you next time.